Hi everyone. For this video, we're going to continue talking about the immune system. In our prior video, we went over NK cells, and now we're going to focus in instead on something called cytotoxic T cells. So just separate that off as part of the prior video. So cytotoxic T cells, we're also going to call them CD8 plus. And these are part of our acquired immune system, meaning that when they're active, they're not just looking generally for cells that don't belong, they're looking for very specific cells. And they look for cells with specific antigens on them. And antigens are just going to be these kind of small protein fragments on the cell surface that identify a cell. Right? So they're kind of almost like a description. They're these unique attributes of a cell that allow them to be recognized by our CD8 plus cells. So the process for a CD8 plus cell to interact is going to be a little bit more complicated than for an NK cell. So I'm going to draw kind of my three cells that are going to interact together here. Okay. So I start out and I have my CD8 plus here, and this is an APC, which is an antigen presenting cell, which is essentially going to give a description of our tumor cell, which is down here in white. It's going to give a description of our tumor cell to our CD8 plus and let it know what to look for as it's roaming throughout the body. So our antigen presenting cell is going to have a molecule called MHC1 on the surface, which is responsible for presenting an antigen. So I'm going to label A. So this is my antigen, which again is just kind of my protein fragment that identifies my target or cancer cell. My T cell will have a receptor called a T cell receptor that's going to be able to recognize that antigen and then go out and look for cells that have that same antigen present on their surface. So the antigen presenting cell is kind of like a witness that gives a description. To the CD8 plus cell of saying, hey, go look for this antigen. So then our CD8 plus is going to go out. This is kind of like step 2 that I'm drawing down here. And it's going to find a tumor cell that matches that description. So it'll be a tumor cell that has this MHC1 molecule with an antigen present. And my CD8 plus will use its T cell receptor to recognize that. So again, this is my MHC1. We have my antigen labeled as an A. This is again that T cell receptor. It's going to recognize this antigen. So I'll label this step as recognizing the antigen. And it's going to attack that tumor cell because it has the matching antigen present. And it's going to use two different methods to do this. The first is that we're going to release some compounds outside of the cell. These are going to be perforin and granzyme B. We won't go into detail about kind of what these do, but they're essentially going to trigger intrinsic apoptosis or cell death pathways. And then the second way that we're going to trigger cell death is by using receptors. So we'll have something called a Fas ligand that will be present on our CD8 plus. Let me draw that like this. This is our Fas ligand. And then maybe we'll have a complementary Fas receptor. And this is going to lead to apoptosis as well. Except this time it's going to be extrinsic signaling. So we have two different mechanisms of inducing apoptosis, either through intrinsic or extrinsic signaling pathways once we've recognized an antigen that's present, right? And we're going to degrade this tumor cell. And then after the tumor cell's been degraded, antigens from the tumor cell are going to be picked back up by the APC once again. And that's how it's able to present these antigens to the CD8 plus in the future. All right. So just to add two other molecules to kind of this CD8 plus interaction, or actually three other molecules. So maybe I'll start down here with CD8 plus in the tumor. There is a ligand that's present on our kind of CD8 plus and our tumor cell that leads to an inhibitory reaction for the CD8 plus cell. So what I mean by that, the CD8 plus has something called a PD1 receptor. And our tumor cell has something called a PD1 ligand. That sticks up off of the cell. When this receptor and ligand binds, it sends a signal towards the CD8 plus cell that is inhibitory, that prevents kind of like activity of the cell. And this is, again, an inhibitory signal that's going to prevent it from attacking and killing this tumor cell. So this is one interaction that can occur that can kind of inhibit the CD8 plus activity. We also have interactions with our APC that can occur. So on the surface of our APC, we have a ligand called B7 that our CD8 plus cell can interact with. And this interaction can occur through two different molecules. I'll put them both on the surface of my CD8 plus cell. In pink, I have a molecule called CD28. And when CD28 associates with B7, this leads to increased activity of the CD8 plus cell. However, our CD8 plus cell also has a molecule called CTLA4. And when CTLA4 interacts with B7, this leads to decreased activity. This is an inhibitory interaction for the CD8 plus cell. So we have our B7, CD8 kind of positive stimulatory interaction, and then we have two inhibitory interactions that can happen either from the CTLA4 or if we scroll back down from this PD1 ligand. So that's kind of my overview of the CD8 plus cell, how it's going to interact with antigen presenting cells as well as tumor cells.